Gyros Protein Technologies is pleased to be here today and talk to you a bit about how the Gyrolab is being used to boost workflow efficiency and data quality in bioprocessing and immunogenicity. Immunoassay applications are widely used in gene therapy. ELISAs are typically used for a variety of applications, from discovery and development through clinical trials and post-market surveillance in the quantitation of viral vectors, reagent screening for assay development, characterization of reagents such as affinity measurements, and determination of anti-drug antibodies. Product quantification, such as viral vector titer or impurity testing for host cell protein contaminants, are also used. And across all of these applications, an ELISA assay is typically the assay platform of choice. What I'd like to do today is show you how, for an ELISA, the drawbacks tend to be long assay time, 6 to 24 hours, require large sample volumes, including large reagent volumes, and often have limited dynamic range. On the Gyrolab platform, we can accomplish the work in about an hour, rapid time to results, use much smaller sample volumes, including less reagent volumes, and the accelerated workflows, which lead to rapid assay development, rapid return of data. So the advantage of the Gyrolab platform is it's a nanoliter-scale immunoassay. What we've done is we've miniaturized the immunoassay and put it on the surface of a compact disc-shaped assay plate. So if we start with the left-hand side of the image here, where we have a streptavidin-coated bead, we use this to build our immunoassay on the surface of this bead using a biotinylated capture reagent. This, in conjunction with an Alexifor labeled detection reagent, can be used to build an immunoassay for any analyte we're interested in. What's unique about the Gyrolab is we've incorporated this biotinylated streptavidin-coated bead in a 15 nanoliter affinity capture column. That column is then built into a microstructure where we have one microstructure as the equivalent of a well on a microtiter plate. So we have one microstructure with reagent channels and sample volume chambers, which direct the liquid flow through the CD and across that affinity capture column at a specified time. Our CD is organized in 8 microstructures per segment. So each segment is combined as a combination of 8 microstructures connected by a single common channel. So on our surface of our CD, we have 12 to 14 segments. So we might have as few as 96 data points or as many as 112 data points on a single CD. When the assay is complete, it gives us a response range, a broad dynamic range to our assay, and it allows us to actually interrogate individual data points by looking at these column viewer profiles where we can see the retained fluorescence on the column. So this microfluidic design eliminates pipetting errors. Individual nanoliter volumes are precisely defined, and samples are processed in less than an hour. Well, while immunoassays are used broadly in all of biotherapeutic drug development, in particular, they can be used in gene therapy and often quantification of things like viral vectors such as AAV or lentivirus, antiviral, anti-vector, or anti-transgene antibodies that could be the equivalent of an anti-drug antibody in gene therapies. For bioprocess, we want to look at AAV titer or P24 titer. And we also want to look at impurity testing in a relevant cell line, such as the HEK293 cells. John Dawson, who's a chief executive officer of Oxford Biomedica, has said that Oxford Biomedica is aiming to develop breakthrough gene and cell-based medicines that improve the lives of patients. And they think that the Gyrolab Explorer is essential as they work towards achieving those ends. So what I'd like to talk about first is a kit that we've developed, which allows you to quantify lentiviral P24. So P24 is a cell surface protein on the capsid of the lentivirus. Measurement of this protein is a surrogate measure of your ability to measure the amount of a lentiviral vector that's present in either a culture media or in a patient sample. So production and development monitoring usually requires the analysis of a large number of samples using ELISA, and so this can be automated by using the Gyrolab system. So microfluidic assay consumes very low sample volumes, and also the critical reagents that are consumed is very small as well. So P24 kit comes complete. It's optimized for the Gyrolab system. It allows you to measure a physical titer measured in viral particles per mil. It includes all the reagents and consumables needed, 96 data points on one CD, or it comes in packs of 50 CDs for a total of 4,800 data points. So the kit performance is very good. We get a relatively broad dynamic range, about 3.5 logs. Again, this uses our biotinylated streptavidin-coated bead. We have our streptavidin-coated bead, which functionalized with the biotinylated capture reagent, which is a monoclonal anti-HIV-1 antibody. This, in conjunction with monoclonal anti-HIV-1 that's been labeled with a fluorophore, allows us to detect the recombinant P24 in this assay. You can see on the right representative curves from 6 separate runs, and the data quality is very good, lays right on top of each other. Again, very nice broad dynamic range. So that's the P24 assay, and this comes as a complete kit that you can obtain off the shelf, ready to go from Gyros. Next, I'd like to talk about the measurement of AAV2. So the adeno-associated virus is frequently used as vector-based therapeutics, and the AAV2 quantification can be automated by using the Gyrolab system and customer-specific reagents. So the assay, again, consumes very low sample volumes and very low critical reagent quantities. In this case, what we've done is transferred a PROGEN assay from the PROGEN plate-based format to the Gyrolab assay. So in this case, we have the A20R antibody that's used as capture and detect in this system. And what we've done is taken and biotinylated one A20R antibody and used that as the capture reagent, and then Alexaflor labeled the A20R to make a detection reagent. So same setup as they have in the plate-based ELISA, we've just adapted it for use in the Gyrolab. The resulting immunoassay has a very broad dynamic range and excellent sensitivity. We get about 3.5 logs of linear dynamic range versus less than 2 logs that are obtained with the Gyrolab or with the ELISA assay. What this allows for is analysis of neat samples versus dilutions that are required to hit this relatively narrow dynamic range from the ELISA. So in this case, the goals were achieved. We were able to transfer the assay from a plate-based system, the PROGEN ELISA system, to the Gyros system, and it allowed us to do this with about 6 runs performed over 1.5 days to have the assay up and ready to go. Sample volumes of less than 10 microliters on the Gyrolab versus 100 microliters on the ELISA, so a dramatic reduction in both sample volume and critical reagent volume. The assay precision and reproducibility was maintained across the platform within acceptance criteria, and the Gyrolab expanded the analytical range, where less than 2 logs of linear dynamic range on the ELISA versus 3 logs on the Gyros assay. So what this enabled then, of course, is a neat sample analysis versus the dilution that was required on the ELISA assay. The last case study I'd like to turn to is improvement of a HEK or HAC293 HCP analysis using the Gyrolab technology. Host cell protein analysis is important because we want to be sure that there aren't any of these contaminants that are riding along in the therapeutic after we've produced the therapeutic in a host cell line. And so in this case, we want to be sure that we want to transfer the assay from using in-house polyclonal antibodies that were available and determine the performance in-process samples from various purification steps. So the format of this assay, again, is a biotinylated capture antibody in conjunction with an Elexiflor labeled detection antibody, which is used to measure the host cell protein contaminants. Performance of the assay, we're looking at 293 in the Gyrolab assay versus the ELISA, and looking at dilutional linearity of several samples across the various process steps from harvest cells through several steps 1 through 6 in the purification process. What you notice again here, looking across both assays, the ELISA on the left and the Gyros on the right, is that for the harvest and the first couple of steps, we see pretty good performance, and the performance is very similar between both the Gyros assay and the ELISA assay. We notice that when we get to steps 3 and 4, we're seeing a slightly elevated sample, but again, dilutional linearity across both sample sets is equivalent. And then what you really notice here is that we're seeing a fall off at the steps 5 and 6 as the sample concentrations get low, and probably in the low range of the assay, the dilution linearity falls off for the ELISA, whereas we were able to maintain the ELISA performance, or the assay performance, across various dilutions for steps 5 and 6 in the Gyros assay. So when we look at these in a graphic form, you can see how the samples tracked with one another. Again, you can see steps 1 and 2. The samples give very similar results, slightly higher results for 3 and 4, and we were able to measure 5 and 6 on the Gyros platform, whereas on the ELISA they kind of dropped off because of the low quantity. So what I hope I've showed you today is that we were able to obtain high quality data with a broad dynamic range, excellent precision and reproducibility. We've automated our assay so that it allows you to transfer assays from plate-based ELISAs with improved or equivalent performance. Faster turnaround times, you can get data back in about an hour versus 4 to 6 hours for a typical plate-based ELISA, and dramatically reduced sample volumes, where in this case we can reduce not only the sample volumes but the reagent volumes required to run an ELISA assay. So with that, I'll take any questions that you have in the room. I really appreciate your attendance and your attention, and look forward to continuing the conversation. Thank you.